closely at a cross-section of human skin. Past the barrier of the epidermis, down in the dermis, lies the infrastructure that keeps us alive. Embedded in this dermal layer is the sudoriferous, or sweat, gland network. This network relies on two distinct tubular structures. Clinically, we classify these as the eccrine glands and the apocrine glands. Under low magnification, their coiled anatomy appears nearly identical. In reality, they execute completely divergent biological tasks. The first system, the eccrine gland, operates from birth as a critical thermoregulator. The second, the apocrine gland, operates as a specialized chemical signaler triggered strictly by emotional response. To accurately map the integumentary system, we will evaluate these glands through a direct, side-by-side comparative matrix. We will assess them across three specific axes: anatomical structure, secretory output, and autonomic control. Conflating these two systems obscures the actual mechanisms behind autonomic responses. Distinguishing them on a structural and chemical level is required to understand how the body manages different categories of physiological stress. We begin our matrix with distribution and anatomy, placing the eccrine system on the left, apocrine on the right. Eccrine glands are distributed ubiquitously throughout the human body. Their highest concentrations, however, are found specifically across areas of thick skin, like palms and soles. Looking at the medical literature, this text confirms eccrine glands are distributed throughout the body, with a dense population over thick skin. Structurally, the eccrine sweat gland is a tubular coiled gland. It consists of two main parts, starting with a coiled portion lying deep within the dermis. This deep coil is built from a single layer of columnar or cuboidal epithelial cells. These are the active secretory units. These secretory cells are physically supported by myoepithelial cells, which interpose themselves within the structure to provide physical integrity. The second structural part is the duct, which passes up through the dermis and epidermis. Unlike the coil, this duct is formed by exactly two layers of cuboidal epithelial cells. Moving to the right side of our comparative matrix, apocrine glands are not ubiquitous coils. They are heavily modified structures. They are geographically restricted to specific anatomical regions, acting as the foundation for the glands of the eyelids, the external auditory meatus, and the mammary glands. This source document notes exactly this, defining the glands of the eyelids, external auditory meatus, and mammary glands precisely as modified apocrine glands. Our first axis reveals clear anatomical boundaries. Eccrine glands are structurally ubiquitous to ensure total body coverage, whereas apocrine glands are structurally modified and geographically isolated to execute localized biological tasks. Our second axis of comparison evaluates the distinct chemical output produced by these structures. The literature describes the eccrine gland's primary output as a clear, watery sweat. This chart details the exact chemical composition of that output: water, sodium chloride, urea, and lactic acid. These glands are fully functional from birth. Their secretion scales up in direct response to rising body temperatures, as well as emotional conditions. This high-volume watery output serves a primary physiological role, regulating the body's core temperature through evaporative cooling. The text for apocrine secretions shows a sharp contrast. Their output is physically viscous, described here as thick and milky. This brings up a diagnostic fact. At the exact moment of secretion, apocrine output is entirely odorless. Human body odor only develops after the fact, when localized skin microorganisms begin growing in and breaking down this viscous apocrine substrate. Because of this chemical makeup, apocrine output plays absolutely no role in temperature regulation. Its production increases strictly during emotional conditions. Within this milky output, the apocrine glands secrete a specific class of chemical substances, known as pheromones, and unlike the lifelong eccrine system, apocrine gland function has a finite biological life cycle, gradually declining in advanced old age. The output data provides a clear distinction. Eccrine glands excrete a watery physiological coolant required for survival. Apocrine glands produce a viscous, emotion-driven chemical signaling substrate. We arrive at the final axis of our comparison, the underlying autonomic control pathways governing these systems. Returning to our eccrine anatomical model, we can trace its specific neural wiring. These glands are hardwired for direct nervous control. They are supplied by sympathetic postganglionic cholinergic nerve fibers. The medical documentation confirms this precise pathway. specifying that the eccrine glands are under the control of these sympathetic postganglionic cholinergic nerves. Mapping the neural pathway for the apocrine system reveals a sharp structural contrast. Sympathetic nerve fibers innervate the surrounding area, but they do not directly trigger the secretory cells. Specifically, these are sympathetic adrenergic nerve fibers. This presents a physiological paradox. Despite this adrenergic innervation, the actual secretory activity of the apocrine gland is not under direct nervous control. The primary text is explicit on this point. It clearly states the secretory activity itself is not under nervous control. Instead, the apocrine system relies on a systemic hormonal pathway. Secretion is triggered by washes of adrenaline traveling through the bloodstream. This adrenaline is released systemically from the adrenal medulla, activating the glands from afar rather than through direct local wiring. The divergence between these systems lies in their triggers. The eccrine system utilizes direct cholinergic nerve signals for on-demand cooling, while the apocrine system responds to systemic adrenergic hormonal washes for emotional signaling. We can now step back and look at our completed comparative matrix. Under structure, we contrast the ubiquitous tubular coils of the eccrine network against the highly localized, modified architecture of the apocrine system. Under output, we track the clear, thermoregulatory water and sodium chloride against a thick, milky, pheromone-rich secretion. And under control, we see direct cholinergic nervous wiring contrasted with indirect, adrenaline-driven hormonal cascades. Memorizing these exact boundaries is vital. Predicting a patient's response in clinical pharmacology or mapping physiological stress responses requires treating them as entirely separate entities. The human integumentary system utilizes two parallel chemical pathways: cholinergic signals for physical thermoregulation and adrenergic signals for chemical communication.